Now we will study the crossing over of chromosomes during the process of meiosis. It must be borne in mind that crossing over of chromosomes is highly regulated process. Now what is crossing over? Crossing over क्या है? The exchange of chromosomal segments between two non-sister chromatids. जो नॉन सिस्टर क्रोमेटिड्स हैं ये होमोलॉग में नहीं है लेकिन नॉन सिस्टर क्रोमेटिड में द क्रॉसिंग ओवर occurs. Just like this, that the exchange of genetic material between homologous chromosomes as shown in this diagram. You see red and blue, they twine together, they attach to the certain points, and the information from the DNA is combined together and exchanged. So the new segments, they are produced. This is we call the crossing over. The basic of crossing over is that crossing over occurs at one or more points along adjacent homologs during synapsis. And the points of contact between each other, the DNA is exchanged from these chromatids, as we saw in the previous picture. Now, in this case, we can see that how these chromosomes, they are joined together, they cross together. You see there are red and blue, and in the first process, the first mitotic division, the two new cells or two new chromosomes are generated, and the second one, again the crossover occurs, and we get 4 different types of the chromosomes. Crossing over is highly regulated. And why it is regulated? Because the crossing over, it occurs at least once for each pair of homologous chromosomes. Homologous chromosomes क्या होते हैं? Homologous chromosomes are those chromosomes which are similar and during the mitotic prophase to ensure the proper segregation, ताकि उनकी proper एक segregation हो जाए of chromosome at the first meiotic division. तो first meiotic division में why it is there, what are the distinct function, इसके क्या फ़वायद हैं of the meiosis? फ़वायद ये हैं कि it helps to hold the homologs together so that they properly segregate. पहले उनको इकट्ठा रखता है ताकि वो एक मौजू वक्त पे और प्रॉपरली वो सेग्रीगेट हो जाएं एंड द टू डॉटर सेल्स, ताकि 2 डॉटर सेल बन जाएं. It contributes to the genetic diversification. Mind you, the genetic diversification is very important for the survival. And the genetic diversification of the gametes, that is the sex cells which are eventually produced. Now, crossing over is regulated by the following ways. The number and the location of the double strand breaks along each chromosome is always controlled. That the double stranded जब break होता है तो this number is controlled and this break will be converted into a crossover. Mind you, then when the double stranded, it opens up, it joins together and make a crossover to transfer the genetic material. The result of this regulation is that each pair of human homolog is linked by about 2 or 3 crossovers in the humans, as we have seen in this picture, where the crossover between the homologs in the human testes has been indicated. You can see there is a crossover, there are the white dots, red dots, and they are crossing over to transfer the genetic material. Now, at least 2 kinds of regulation that influence the location and the number of crossover formed. Both operate before the synaptonomal complex which assemble. इससे पहले ये crossover होना ज़रूरी है. And one ensures that at least one crossover form between the member of each homolog pair as is necessary for normal homolog segregation in meiosis 1. ये क्यों ज़रूरी है? ताकि genetic diversity जो है for the next generation it should be continued.